Over the last few lectures, we've discussed in some detail how the chemistry of life works. Specifically, we've looked at the energy relationships of different kinds of biochemical reactions, contrasting anabolic reactions that build more complex molecules out of simpler ones, but requiring energy to do so, with catabolic reactions that break down more complex molecules and release energy. We've also talked about the role of ATP as a fundamental energy currency in the cell, and how ATP can donate the energy stored in the bonds of its phosphate groups to drive anabolic reactions energetically uphill, thus allowing cells to perform all of those processes necessary to synthesize the biomolecules living things are made of. Finally, we've introduced the idea that cells can potentially recharge ATP by adding back a phosphate group to ADP. Now, I suggested that the energy needed to recharge ATP in this way could be captured from the energy that's released when catabolic, those are exergonic reactions, break down organic molecules. We referred to this breakdown of organic molecules as oxidation because the way that energy actually is released has to do with the transfer of electrons from the chemical bonds that are more closely associated with carbon atoms, a relatively non-electronegative element, to bonds that are more closely associated with oxygen, which is highly electronegative. Now, the trick, we said, is to release the energy produced by these catabolic reactions in a controlled fashion, in a way that allows the cell to capture it. Remember, oxidizing organic molecules is chemically the same thing as burning them. But if we burn an organic compound, the energy is released in a sudden, uncontrolled fashion, and it's lost as heat, and therefore it can't be captured and used by the cell. In today's lecture, we're going to build on this background and begin to look at those metabolic processes that have evolved to capture energy from the breakdown of organic molecules in a controlled fashion. We'll focus our discussion on the breakdown of the sugar glucose, because in fact this is the most common organic fuel that's oxidized to produce cellular energy. We'll note later, however, that all sorts of other organic molecules also can be thrown into the mix and provide fuel for making more ATP. Now, the complete breakdown of glucose to release the energy stored in this molecule involves three distinct sets of metabolic processes. These processes are referred to as glycolysis, the Krebs cycle, and a process referred to as the electron transport chain. Now, taken together, the Krebs cycle and the electron transport chain are referred to as cellular respiration because, as we'll see later, these processes require the presence of oxygen to run. Now, in common use, people sometimes refer to all three processes of glycolysis, the Krebs cycle, and electron transport as cellular respiration because most cells use the products of glycolysis as the starting point for these other two sets of reactions. This is really a detail we don't need to be concerned with, but I might say cellular respiration in a more general way, or I might refer to it just in terms of the Krebs cycle and electron transport. Now, let me begin by providing you an overview of what each of these three processes does for the cell to provide kind of a roadmap to see where we're going in the next few lectures. After I've given you this overview, we're going to go back and look at each process in a little more detail in turn. Glycolysis is a catabolic pathway that begins the breakdown of glucose, which you'll recall is a 6-carbon sugar, by breaking it down into two 3-carbon compounds called pyruvate. Glycolysis is a metabolic pathway that actually includes 10 steps and thus 10 different enzymes to run. The reactions of glycolysis occur in the cytoplasm of the cell. In other words, the enzymes that mediate these reactions are found in the cytoplasm, and this is where glucose is broken down to form pyruvate. Now, as we might expect, the breakdown of glucose to form pyruvate is exergonic. It's energy releasing overall. And some of the energy that's released by glycolysis is harvested by the cell. It's harvested in two ways. First, the cell produces 2 molecules of ATP for every glucose molecule that's split in two. This is done through a process that we're going to call substrate level phosphorylation. We'll come back to this point later on, but in short, substrate-level phosphorylation refers to the involvement of some sort of coupling enzyme that directly couples the energy released from one of the reactions of this metabolic pathway to the production of a molecule of ATP. Second, the cell also captures some of the energy that's released when glucose is split in the form of electrons that are captured by the electron carrier NAD+, essentially reducing this NAD+ to NADH. Now, remember that NAD is one of those two electron carriers we talked about last time that's able to pick up electrons because it's somewhat more electronegative than the substance it's interacting with, and it does so through what we call the dehydrogenase enzyme. So the cell does get some energy out of glycolysis in the form of a couple of molecules of ATP and some electrons that are stored in NADH. Now, we'll see later, actually, that the energy in these electrons in NADH is not really directly useful to the cell and has to be converted in a different process to form ATP. Now, interestingly, we've broken glucose down, this high-energy molecule, and we have captured some energy, but we really haven't captured very much energy. In fact, there's still an enormous amount of energy that's left in the carbons that made up that glucose, and this energy is in those pyruvate molecules, which are the end product of glycolysis. Another way to put this is that the carbons in pyruvate are still highly reduced. They're still in a fairly reduced high-energy form. Even more energy could be extracted from these carbons if we could somehow further oxidize them. Well, it's the Krebs cycle, this second set of metabolic processes that completes the job of breaking down pyruvate, completely oxidizing the carbon in it. And in this way, extracting as much energy as possible from the glucose molecule. In fact, as we'll see, most of the energy that a cell harvests from glucose is actually obtained through the Krebs cycle as compared to glycolysis. Now, the Krebs cycle is named for Sir Hans Krebs. He was a German biochemist. He fled Germany in the 1930s and worked in England. And in the 1930s, he actually worked out most of the details of this metabolic process. And in fact, this work led him to being awarded the Nobel Prize in 1953. Now, like glycolysis, the Krebs cycle also represents a series of reactions in a metabolic pathway. In this case, there are 8 metabolic steps, each mediated by different enzymes. Now, as we'll see, this process actually is called a cycle because the metabolic pathway loops back on itself. In other words, the last compound in this pathway actually forms a substrate for the first reaction. So the compounds go around in a circle, essentially, which is why we call it a metabolic cycle. The significance of this point will become more clear when we actually talk about the Krebs cycle in more detail. Unlike glycolysis, the reactions of the Krebs cycle don't occur in the cytoplasm of the cell, but instead occur inside an organelle known as a mitochondrion. We were introduced to mitochondria, that's the plural for mitochondrion, at the very beginning of the course when we first talked about the structure of your eukaryotic cells. This is the organelle that's specialized for processing energy, one of the 2 organelles. We'll learn about another one in a few lectures. Because the Krebs cycle occurs in the mitochondrion, the cell actually has to transport pyruvate, the end product of the Krebs cycle, of the glycolysis, into the mitochondrion before the pyruvate can be further oxidized. Now, as the Krebs cycle completes the oxidation of pyruvate, it produces carbon dioxide, that's CO2, as an end product. CO2 is the most oxidized form of carbon. This is because the carbon atom is bound to 2 oxygen atoms, and therefore the electrons associated with the carbon are held more tightly by the oxygen. And remember, oxygen holds its electrons in very tightly, not allowing them to carry much energy, and therefore we say that the carbon is highly oxidized. The complete oxidation of pyruvate by the Krebs cycle produces more ATP through this process of substrate phosphorylation. Remember, what that means is that some of those reactions in the Krebs cycle release energy, and this energy is used, this energy is coupled to the production of ATP by a coupling enzyme. That's what we're calling substrate level phosphorylation. However, the vast majority of energy that's released from these carbons in pyruvate, from glucose that's released through the process of the Krebs cycle, or I should say captured through the process of the Krebs cycle, is actually captured in the form of more electrons that are picked up by electron carriers, the electron carriers NAD plus and FAD. Now, the energy that has been captured from the Krebs cycle and glycolysis in the form of ATP is immediately usable by the cell. ATP is the currency that runs cellular processes in general. But the energy that's been captured in the form of the reduced forms of these electron carriers, which would be NADH and FADH2, that's not an important detail, but the reduced forms of what we'll just call NAD and FAD, this energy isn't actually very useful to the cell. Even though the electrons that have been captured have a lot of energy associated with them, the cell can't use that energy to do anything immediately useful. Instead, it has to take that energy and somehow convert it to the usable form of ATP. The third process I mentioned, the electron transport chain, is the process that accomplishes this task, extracting the energy stored in NADH and FADH2 and using this energy to make ATP. The electron transport chain the electron transport chain does this through a unique process, actually a process that was quite mysterious for some time, called oxidative phosphorylation. In fact, sometimes we use those two terms interchangeably. We might talk about the electron transport chain, or we might just simply refer to oxidative phosphorylation. Now, the difference between substrate-level phosphorylation and oxidative phosphorylation is something that we're going to talk about in more detail in a couple of lectures, but let me just give you an idea of where we're going with this. As I said, substrate-level phosphorylation involves the direct coupling of energy release from an exergonic reaction to make more ATP with the help of a coupling enzyme. In oxidative phosphorylation, the cell has first captured energy from a redox reaction that transfers electrons to an electron transport molecule, an electron carrier. And then the electron carrier takes those electrons to another set of molecules, gives up those electrons that the electrons it's carrying to those molecules through another redox reaction, and then there's a series of redox reactions whereby those electrons are stepped down gradually in energy level with the release of energy at each of these steps. Eventually, those electrons will end up associated with oxygen atoms, but only after they've gone through a long series of intermediates. As I said, how oxidative phosphorylation actually works was a mystery for many years, and we're going to talk about that later. But in a nutshell, what's happening here is that the redox reactions of the electron transport chain are powering the pumping of an ion up its concentration gradient, and this concentration gradient has now some energy stored that in a separate process will be used to drive the phosphorylation of ADP to make ATP. Now, there's one more general point I want to make about these processes, and that is that the Krebs cycle and electron transport only function if there's oxygen present in the cell to serve as an ultimate electron acceptor at the end of the electron transport chain. Now, most cells, I should say cells of most organisms, accomplish cellular respiration in the presence of oxygen and therefore carry the breakdown of glucose all the way from glycolysis through the Krebs cycle and through oxidative phosphorylation. We call this aerobic respiration, aerobic in biology simply referring to any process that requires oxygen. Some organisms, however, only accomplish respiration in an environment that lacks oxygen. Furthermore, sometimes cells that normally perform cellular respiration in the presence of oxygen find themselves in an environment that is lacking oxygen or doesn't have enough oxygen to accept all the electrons that it might. In these cases, cells can only perform the reactions associated with glycolysis. That is, they can only take the breakdown of glucose as far as the production of that 3-carbon molecule pyruvate, which is the end product of the glycolytic pathway. Anaerobic respiration recovers much less energy from glucose. In fact, as we'll see, only 2 molecules of ATP are produced by the breakdown of a glucose molecule. Aerobic respiration is much more efficient because it extracts almost all of the energy stored in glucose. Well, not all of the energy, but as much energy as can possibly be extracted, completely oxidizing the sugar. Okay, that's the big picture. Those are the three reactions that we want to look at in three sets of reactions that we want to look at in some detail. Now what I'd like to do is to look at the first of those sets of reactions, glycolysis, in a little more detail. Glycolysis literally means splitting sugar. Glyco, sugar, lysis, breaking in two. The glycolytic pathway, the reactions associated with glycolysis, start with a molecule of glucose as the substrate, and this is a 6-carbon sugar, and as I said, they end up with two 3-carbon molecules called pyruvate. Now, the structure of pyruvate is just a simple chain of 3 carbon atoms with 2 of these carbon atoms double bonded to an oxygen and the carbon on one end bonded to 3 hydrogen atoms. Actually, we don't need to worry about the details of the chemical structure here, but one point to make is that you can guess that pyruvate is more oxidized, or the carbon in pyruvate is more oxidized as compared to glucose because there's more oxygens associated with those carbons. As I said earlier, in the process of splitting the 6-carbon glucose in two, we've partially oxidized some of the carbons in that molecule, releasing some of the energy that was stored. Now, there are actually 10 separate reactions involved in the glycolytic pathway, with each of these reactions catalyzed by a different enzyme. One way we could look at glycolysis, actually, would be to run through the process step by step, looking at every one of those reactions, looking at every enzyme, the substrate it works on, and the product it produces. For example, the first reaction of glycolysis is catalyzed by an enzyme called hexokinase. Hexokinase is an enzyme that acts on glucose as a substrate and yields a modified form of glucose called glucose 6-phosphate. Actually, it's added a phosphate group to this glucose. The second reaction is catalyzed by another enzyme, phosphoglucoisomerase, which yields a product called fructose 6-phosphate, and so forth. You can imagine this could be a lot of biochemistry to keep track of, and it would be very easy to get lost in the details of all of this. Fortunately for us, we can get a pretty good idea of the key issues we need to understand about glycolysis without going into this level of detail. Instead of worrying about the details of each step, what we want to do is focus on energy. Focus on changes in the energy levels of the intermediate compounds that are produced as we move through glycolysis without worrying about the names of either those compounds or the enzymes that catalyze them. Now, I'm going to throw in a name or two along the way when I describe a little bit about this, but we don't need to worry about those. I'll just throw in a few of the key players to give us some sort of points of reference. Let's go back to thinking about changes in energy level in reactions in terms of a graph that has on its x-axis time, that is the progress of the reactions we're interested in, and on its y-axis the relative amount of free energy in the compounds involved. Now let's imagine we're going to represent the reactions of glycolysis on such a graph. We don't need to add names to our graphs. What we do is we just mark the relative level of energy possessed by each of the intermediate compounds, essentially the 10 compounds that occur along the pathway. Each reaction in the pathway, because it converts one compound to another, will likely change the relative energy level. That is, what we'll see is as we move along the x-axis, there'll be sort of step-like changes in the relative height of the line, representing changes in relative free energy. Now, one thing we do know about what this graph would look like is that the very beginning of the pathway, the molecule glucose, would be at a relatively high energy level as compared to the very end of the pathway, the molecule pyruvate. We know this because we know that glycolysis, the pathways associated with glycolysis, are in aggregate, exergonic, that we are giving up energy by breaking glucose down into the 3-carbon pyruvate molecule. However, if we graphed out all 10 of the steps between glucose and pyruvate, we'd notice something that's a little bit less expected. Specifically, the first 5 steps in the glycolytic pathway actually go uphill energetically. In other words, these first 5 reactions don't step glucose down to lower energy levels, but in fact step it up to higher energy levels. Now, the energy that's needed to drive these reactions uphill comes from, you might guess it, ATP. In fact, to get these reactions, the first 5, to run uphill, 2 different molecules of ATP come in at different places, donating their phosphates, and in so doing, raising the energy level of the molecule. Now, this is surprising. What glycolysis is supposed to be doing is to give us energy. But what we're doing now is we're putting energy into it. What's going on here? This seems counterintuitive. Well, the reason that the cell needs to invest some energy at the beginning of the glycolytic pathway really has to do with the fact that the molecular structure of glucose needs to be rearranged into a configuration that will allow it to then be stepped down energetically later on in a more controlled fashion. Remember, the cell needs to have energy released in small steps, not all at once. If we just oxidized glucose, what would happen would be a very enormous release of energy. So the cell has to invest some energy to basically rearrange the bonds in glucose a little bit to get it into a shape where it can then control the remaining reactions. We refer to these first 5 steps, the first 5 upward steps of glycolysis, as the energy investment phase of glycolysis. Now, once energy has been invested in this 6-carbon sugar and its molecular structure rearranged a little bit, an enzyme called aldolase comes in and splits that 6-carbon molecule in half, creating 2 3-carbon sugars. These 3-carbon sugars are now occurring at the highest energy level. These sugars are called glyceraldehyde 3-phosphate. Or we could call it G3P for short. It's a lot easier to say. Now, glyceraldehyde 3-phosphate is quite a mouthful of a name, but it's one of those names that I think is worth remembering, not only because it's the energetic high point of the glycolytic pathway, but also because this is a molecule we're going to encounter again when we talk about photosynthesis a few lectures down the line. It turns out that G3P, glyceraldehyde 3-phosphate, is in fact the 3-carbon sugar that's manufactured by photosynthetic organisms when they convert sunlight into more reduced carbon molecules. Now, the steps of glycolysis that follow the production of G3P are mostly exergonic. They mostly then run downhill from that point. And so we refer to the last 5 reactions of glycolysis as the energy harvesting phase, or the energy yielding phase of glycolysis. In fact, the very first step of this energy-yielding phase of glycolysis is a highly exergonic reaction. It's so exergonic that it not only reduces a molecule of NAD+ to produce NADH, in other words, it transfers an electron from that molecule that's being oxidized over to one of our electron carriers, it also releases enough energy to add yet another phosphate to this molecule. Now, we've got a couple of phosphates that have been attached in this energy investing phase already to the carbons that we're working with. We've got another one that's attached to each one of these 3-carbon molecules that's going to go down the line energetically. These phosphates that are being attached to this molecule will then be donated through exergonic reactions later on down the glycolytic pathway to ADP through substrate-level phosphorylation to create some ATP. Okay. The second step of this energy harvesting phase, the second reaction, also is very exergonic, and this is where some of that ATP is produced through substrate-level phosphorylation. The next couple of reactions after that don't change the energy level too much. They're just a few rearrangements of the molecule. But then there's another highly exergonic reaction a little further down the line, which yields yet another molecule of ATP, again, by one of the phosphates that's attached to this carbon that's being oxidized, to being transferred to ADP through substrate-level phosphorylation. What we're left with at the end of these 10 reactions, at the bottom of the energy-yielding phase of glycolysis, is that 3-carbon molecule pyruvate, which is relatively oxidized at a lower energy level relative to the glucose that we started with. Well, all right, so these are a lot of biochemical reactions we've just run through. What has the cell gained in terms of energy? Because that's what we're interested in, what the cell gets out energetically. Well, we had to invest a couple of ATPs early on in glycolysis. So we're already sort of negative when we start counting up how much energy we get. We've had to spend something. But in the energy yielding phase of glycolysis, we actually get a total of 4 ATP molecules back. So overall, through the whole process of glycolysis, we've actually yielded a net increase of 2 ATPs being produced for the cell. These ATPs actually only represent about 3% of the energy that was originally stored in glucose, so we haven't gotten very much energy out yet. We've also gotten some energy, though, harvested in the form of these electrons that were transferred to electron carrier molecules. But as I said earlier, those electrons, or the energy they carry, are not directly usable by the cell. Something else has to happen with them later on. Now, there's one more point I want to make about glycolysis as a metabolic pathway. And that is that the production of NADH, that's the reduced form of the electron carrier molecule that's taking electrons away from some of the carbons as they become oxidized, the production of NADH actually presents the cell with another challenge. Specifically, the cell needs the oxidized form of this electron carrier, the form that doesn't have electrons, to pick up electrons. In other words, this oxidized form of NAD+ is, in a sense, a substrate of one of the reactions of glycolysis. What that means is that if there isn't enough NAD+ to go around, glycolysis can't run. That first energy-yielding reaction of glycolysis has to give up electrons to something. And if there's nothing there to accept them, then glycolysis, the whole pathway, comes grinding to a halt. And it turns out that in general, the supply of NAD+, the oxidized form of this electron carrier, is a limiting factor in cells. Cells do run out of NAD+. If glycolysis runs too much, and if that NAD+, or the NADH that's produced, doesn't somehow get rid of its electrons somewhere else, then the process stops because no more electrons can be picked up. Well, there are two kinds of processes that can actually recycle NADH. These processes are associated with reactions that we generally call fermentation. Fermentation doesn't actually gain any more energy for the cell directly. All it's doing is recycling these electron carriers. If they can't donate their electrons to anything useful, it recycles these electron carriers so that they can go and pick up more electrons. Now, fermentation then occurs in the absence of oxygen, because as we'll see later, if oxygen is present, then these electron carriers will give up their electrons to the electron transport chain, and then they can be recycled that way. But if an organism is doing anaerobic respiration, if an organism is breaking down glucose in the absence of oxygen, then it has to get rid of those electrons in some other way, and the processes of fermentation are how it does it. There are actually two main types of fermentation, and they're worth pointing out here because actually, from a human point of view, they've proven to be quite useful over time. One of these processes we call alcohol fermentation. In alcohol fermentation, what happens is that electrons are transferred from NADH back to pyruvate in one form, and in this case, the pyruvate is converted to ethanol, a kind of alcohol. We're most familiar with alcohol fermentation, which is done by yeast in the context of brewing and winemaking. Many bacteria also use alcohol fermentation when they're just lacking oxygen. Now, the second kind of fermentation, the second class of fermentation, we call lactic acid fermentation. In this case, pyruvate is reduced. It picks up electrons from the NADH in a single step to produce a different kind of molecule, a molecule called lactate. Now, lactic acid fermentation is done by various kinds of fungi or bacteria that have been used in human culture for the processing of dairy products for thousands of years to make things like yogurts and cheese. Also, we're also familiar with lactic acid fermentation because when we exercise too much, when we exercise vigorously, it's possible for our own muscle cells to run out of oxygen. What happens in our muscle cells then is that only the glycolytic pathways can run, not the aerobic pathways that include electron transport, and we have a buildup of NADH which has to be shuttling its electrons to something else. It does this through lactic acid lactic acid fermentation, causing the buildup of lactate in our muscle cells. This is one of the reasons your muscle cells get sore when you exercise strenuously for too long. When you stop exercising, this lactate then is transported through the bloodstream to the liver, where it's converted back to pyruvate and can be shuttled into the reactions of aerobic respiration when oxygen becomes available again. Well, we've now followed the biochemical pathways that extract energy from glucose as far as the end of glycolysis, and we've gotten out of that just a little bit of energy that the cell can use. In the next lecture, we're going to pick up this trail again and look at the rest of the processes begin to look at the rest of the processes of cellular respiration, where we'll see the real energy payoff is to be had.